Hello everyone, and in today's video I will be going over exercise 5-10, which is the bile esculin test, and exercise 5-12, which is the urea hydrolysis test. Alright, so before going into these tests, I kind of want to take a step back and look at the big picture regarding the tests that we'll be doing this week. Now the tests here are designed to look for hydrolytic enzymes, which are enzymes that use water in order to break down large complex molecules. Now those tests are then further split up into the first group being the intracellular enzymes and compared to the extracellular enzymes. Now intracellular enzymes are enzymes that are produced within the cell itself, and so all the activity happens inside the cell body. And extracellular enzymes, also known as exoenzymes, are released out into the exterior environment of the cell, and thus activity then occurs outside. Okay, and of course, as I mentioned earlier, these tests are performed and designed to identify the actions of hydrolytic enzymes, and of course, as with many of the other tests, enzymes provide a great way to differentiate large groups of bacteria. All right, so in the first test, being the bile esculin test, you'll be working with two organisms here, L. lactis and Enterococcus faecalis. E. faecalis is actually a BSL-2, so you will need to wear gloves for this test. Okay, so once you have your bacterial cultures, you will then need to grab all the tools that you will need. And alongside that, you will of course need one bile esculin agar plate and one permanent marker. Okay, so once you have your materials with you, we can then go ahead and begin inoculating your bile esculin plate. But before you do that, you want to properly label your plate. And so the first thing you're going to do is flip the plate upside down, and using your permanent marker, just draw one line across that will split the plate in half. And of course, in this region here, you will inoculate it with L. lactis, and the second half you will inoculate with E. faecalis. And what we'll then do, of course, is you have to prepare your tools by sterilizing them. And once you have done that, you can then bring the organism over onto your bile esculin plate. And what you'll do is just grab a little bit of that culture there and just perform a simple spot inoculation, making sure that the organism that you are using is the one that has been assigned to that specific half. And then, of course, you will then repeat that once again with your second organism. Okay, so moving on. When you come back, you will see something like this, where one organism here has grown, and the other one also has grown successfully, but it is covered in this black precipitate. And so what's going on here? Why is this black color formation happening here? Well, that is because the main component within the bile esculin plate is esculin. Now, esculin is this complex molecule that is comprised of one glucose molecule that is attached to one molecule of esculitin. Now, organisms that are bile esculin positive, as you can see in this colony here, are able to break down the esculin by cleaving off the esculitin from the glucose. And the esculitin then will interact with this chemical known as ferric citrate, which plays the role of the indicator for the test medium itself. And when these two chemicals interact with each other, it forms this black precipitate, which is indicative of an organism that is positive for or is bile esculin positive. And compared with this colony here, where there is none of this sort of black formation, it is bile esculin negative. Right, so why don't we review real quick what we went over? This test, of course, is designed to look for bile esculin positive organisms, and those are organisms that, of course, are able to produce an enzyme that can break down esculin. When esculin is broken down, esculitin, a byproduct, is formed, and that interacts with the indicator ferric citrate that then generates that black precipitate that tells you that organism is bile esculin positive. Now, in terms of application, this test is used to presumptively identify members of Enterococci and members of Streptococcus bovis. And so this shows you that this is more so a test that is used when you have kind of gone further down in your project and you are sort of at the level of genus and species. Of course, you could perform this test earlier if you wanted to. However, it's just at that point, it really doesn't provide you with any information to kind of broadly remove large groups of bacterial organisms, so you can't really do much with it. All right, so let's move on to the next test, which is the urea hydrolysis test. And for this test, you'll be working with A. faecalis and E. coli. Once again, I'm sure you guys are very familiar with this. You want to then get all of your lab materials ready to go, and alongside 3 urea slants. Now, once you have everything together, you can then proceed to inoculating your slants, of course, after having labeled all of them. What you want to do is just, once again, prepare all your tools to transfer the bacteria itself. And once you have done that, see here, just kind of grabbing some of that organism there. Obviously, your loop cannot actually go through the glass. But yeah, and then what we're going to do is then flame the tip of your urea slant and then perform a simple fishtail streak inoculation. Just kind of going back and forth, creating that zigzag pattern. And then at the tip, you can then, you are then officially done. Okay, so you'll do that for this urea slant here. And of course, this third one here is your control. Okay, so once you're done, once you're done inoculating and you come back and you look at your tests, you'll see that one of the test tubes here has turned into this bright yellow color, while this one here has kind of just maintained the same color it originally had. And in this third test tube here, it has turned into this bright pink color. Now, what's going on here? So we can tell here by the results table that this organism here is urease positive, and in these two test tubes that they are urease negative. However, timing is very critical for this test. After inoculating, you will then incubate this test for a total of 24 hours. However, this test can then be re-incubated up to a total of 6 days, where after every 24-hour interval, the test will need to be checked. And this is because urease positive organisms fall under 2 categories, and I've kind of bolded and underlined here to show that there are organisms that are rapid urease positive, and that there are also organisms that are slow urease positive. Now, organisms that are rapid urease positive have the ability to produce a lot of this enzyme known as urease that can then break down the core substrate known as urea that is integrated within the test media itself. And so because they're able to produce so much of these enzymes, they're able to very quickly break down the urea, which in that process produces a lot of ammonia. And ammonia is a very alkaline chemical. And so as that ammonia builds up, the overall environment within that media becomes very basic, and it's able to kind of push past this buffer that is also included within the test media itself, shifting the color of the phenyl red indicator to turn pink. Now, slow urease positive organisms are not able to kind of push past that buffer and turn the indicator pink within that 24-hour time frame. And that's because they're just not able to produce urease that quickly and, of course, consume all of that urea within the media itself. And so that's why you see there is this distinction here of post 24 hours and or these urease test slants can then be re-incubated for another 24-hour cycle, and potentially you can do this up to 6 days. You can then see that over time this slow urease positive organism can then shift the indicator to turn pink. So in our situation here, we don't necessarily have to worry about that because we are aware of the organisms that we are using. And of course, this being our control here. However, if you are in a situation where, one, you're not even aware of what species you're dealing with, and two, you have a whole range of bacteria that you need to kind of cut out and narrow down, you want to be very careful and try not to jump to the conclusion when you see a test tube that has still remained orange, because potentially that organism may not be negative. It just needs a little bit more time for it to break down enough urea to then, of course, allow that ammonia to build up and change the pH environment so that the phenyl red indicator can then change colors. Now, this last test tube here, of course, we know that this is negative because if the organism had the ability to produce urease, it would not have turned the color yellow. Instead, it would have turned pink. And what we're seeing here is that the organism has just then proceeded to ferment glucose. And we all know that the phenyl red indicator, when it is in an acidic environment, will turn yellow. All right, so I apologize about that. The thing that I want you to kind of take away from this video is the fact that this test is, of course, designed using urea as the main substrate to distinguish between organisms that can produce the enzyme urease versus those that cannot. And of course, the bacteria that do have that ability to produce that enzyme, it is then further separated into two groups where one can produce it very quickly while the other one requires a little bit more time to do so. And finally, that this test is good to use when you know you are potentially dealing with a urinary tract pathogen. I'm sure folks who have taken A&P know that within the urinary tract, urea is a very common chemical found there. And as you have all probably have known, when you do take like a urine test, urea is one of the chemicals that are tested for. So you can see why it's a good environment for these pathogens that tend to infect that general region. So if you know you're dealing with an organism, Proteus being one of the common culprits of urinary tract pathogens, you can use this test to help determine if that is what you're dealing with. All right, so that brings us to the end of our video. In the next video, I will be talking about the last 3 tests that we will be working with for week 10. And those tests will then go over extracellular enzymes. The 2 tests here focused on intracellular enzymes. And so the next 3 tests will kind of show you how those 2 enzymes may differ, and continue our study of the hydrolytic enzyme tests. I hope you guys found this video helpful. I really appreciate you guys. I wish you guys luck in your continued studies, and I will see you guys in the next